Oh, hi. I'm David Lally, and I'm standing in this cornfield at Virginia Tech because I want to introduce you to an experiment you're going to be doing with Dr. John McDowell. You can't see this right now, but I'm surrounded by thousands of disease-causing microorganisms. Those germs are in the air, they're in the soil, and most likely they're on the plant themselves. But when I look at these corn plants, I can't find any evidence of disease. So how are these plants defending themselves against disease-causing microbes? Let's go talk to Dr. John McDowell and see how his research is helping to answer that question. Hi, everybody. Now I'm here in the office of Dr. John McDowell. How's it going, John?
It's going great, David.
How does your research answer that question? I mean, the plants are out there and there's all these disease-causing microorganisms. How do plants defend themselves?
Well, as it turns out, plants are actually really good at seeing when they're being invaded. As you pointed out, they live in an environment that's teeming with microorganisms. But plants have this particular capability for being able to see when a microbe has entered their environment. And when they realize that, then they can turn on really sophisticated and effective defenses. So, for example, one defense that they use is that infected cells can actually destroy themselves. And that can either kill the pathogen outright or at least cut off its nutrient supply.
So really what you're trying to understand is like the plant has an immune system.
Yeah, exactly. And in fact, if you compare the proteins that plants use to recognize pathogens with the proteins that humans use to recognize pathogens, there are a lot of similarities. And in some cases, those proteins are almost identical. So there's a lot of carryover between those two fields.
So even though it looks like this really healthy cornfield, there's this virtual warfare going on between microbe and plant.
That's right, yeah. It's probably safe to assume that at any given time out in the field, some part of the plant is actively defending itself. You can't see it, but there's a lot going on underneath the surface.
And so what you're trying to understand is not just how the plant defends itself against the microbes, but how the microbes evade those defenses.
Exactly. That's the other side of the coin, and Homeland Security is kind of a good analogy in this regard. So the plant is making dozens of proteins that enable it to see when it's being invaded. Okay? And so what the pathogen is doing to get past the surveillance system is to either disguise itself in some way or to short circuit the system that enables those surveillance proteins to turn on defenses once they've recognized that an invader is somewhere in the vicinity.
Awesome. So I'm interested to know how exactly you're going to do that and the experiment the students are going to do. Can we walk over to your lab and have a look over there?
That'd be great. Follow me.
Awesome. Thanks. So here we are in John's lab, and what's that you have there, John?
So this is a pot with Arabidopsis plants.
And how are you using these in your experiment?
What we're doing is we have inserted a gene into these plants that comes from a pathogen that is very good at establishing an infection on Arabidopsis plants. And what we'd like to figure out is, okay, what is this gene actually doing to the plant to make it more receptive to being invaded by this particular type of mildew?
No way. So you put a gene from the pathogen into the plant genome?
Yeah, that's right. And that's the sort of thing that's really easy to do with Arabidopsis, and that's one of the reasons that people like me like to do research with Arabidopsis. Those sorts of experiments are really straightforward and easy to do.
So rather than exposing the plant to the whole microorganism, the whole pathogen, you're just putting one gene that is sort of, signaling the Arabidops maybe that it's under attack.
Yeah, that's right. And we're doing the same experiment for a lot of different genes from the pathogen. And so we're looking at what each gene does one at a time, and when we have results from those experiments, we hope that'll give us a broader view of what might happen when the pathogen itself is interacting with the plant.
So what exactly will the students be doing with these plants?
What we hope the students will be interested in is addressing our hypothesis, which is that this particular gene might affect the plant's ability to resist stresses that are coming from the environment.
Awesome. Can you be more specific?
Sure. So One thing to keep in mind about these pathogens is that their aim is to live inside plant hosts. And so it's conceivable that it might be to the pathogen's advantage to make the plant more resistant to something like drought or high temperature or other environmental variables that might harm the plant's ability to grow. So we're hoping that those are the sorts of things that the prep students might be interested in testing with their own experiments.
So you want to know how the plant might respond differently to those stressors like drought and heat and different light and stuff like that.
Exactly.
Awesome.
Yeah. And this is a great thing for us to collaborate with prep on, because there are lots of different experiments that could be done to test this particular hypothesis, but we're not going to have time to do it. So for us, this is a really great collaboration, and we're really excited about it.
All right. Well, thank you very much, and we'll see you all in the classroom.
See you guys soon.